Log on, tune in, find out. Another good idea from Cambridge.
Our first set released about 2 years ago, published last year. Looked at 180 people. We found 15 million new genetic variants. These are places in the DNA which are different between people. Last year there was an interim, 600 people, 26 million variants, and this year we have already released sort of near final phase 1 set on nearly 39 million genetic variants. So the aim of some sense of this is to try and capture the genetic variation in the population, find places where it's likely, so that if you take any new person, if you look at where they differ from the population, from other people, that we will have a good picture of what that is. Most of those variants have already been found. So there are a lot of kind of words like most in there, which are standard to mathematicians, epsilons and deltas and so on. And we need good, it's sensible, if we're going to do this, to try and understand how we're doing, have good measures of how we're doing, and what designs are strong for achieving this. So on this plot we see, here, there's an international repository called dbSNP of genetic variation. And these are different builds of dbSNP. You can think of them as time. And you can see an increase in the number of variants found. And in fact, these two columns essentially have been added by the pilot in phase 1 of 1,000 genomes. And on the other axis, in blue, are 2 individuals. And these are not people we've sequenced, but as part of the project, we've sequenced them separately. We're asking the question, what fraction of the variation in those people has been found in our pool? And you can see as we've sequenced more, that fraction has dropped. And so for a European, we've got, this is on a logarithmic scale, we've got about 1.5% that's not, we found 99.5% or 98.5%. And for an African, we haven't quite covered them so well. Actually, there's more variation in Africa, it's more complex. And we've got about 98% variation. Okay, so that all seems good, natural measure. But it's a little more complicated than that. What type of variation are we talking about? Well, we found here, there's a specific example. 98% of variants in this person here were found in the pilot data. There's about 3 or 4 million. But 1 or 2% left that are novel in 3 or 4 million is still quite a lot. It's tens of thousands. But it turns out that amongst those tens of thousands, they're enriched to things that we're probably most interested in. So if we look at non-synonymous changes, these are changes which change proteins, which actually change the function of genes. We've only got 95% of those. If we look at ones that actually kill off the protein, that stop the protein, we've only got 88%. And then you can take a person and ask, do they contain mutations that we think cause disease? We've got some evidence. And each person does. None of our genomes are perfect. We all carry things which are disease causing. Luckily, we've got 2 copies, so in most cases that won't be too bad for us. We've only got 85% of that class. So how could that be? How could there be differences? Well, it's because each variant has a frequency in the population, and this is the frequency spectrum from half to 1, it's from 0 to 1. And there are many more rare variants than there are common variants. So, of course, if you've got something that's 50% of the population carries and you sequence 100 people, it's very likely you'll have found that. But if there's something that's got a 1% population frequency, then it's less likely we'll find it. And there's a sort of 1 over x type distribution here. And that distribution is skewed for functional variants. And in fact, the gold line here, so this is relative. These are relative, so flat. This is the average for the whole genome. And non-synonymous ones, the protein-changing ones, are a bit skewed, more rare ones. And for the really functional ones, the stop codons or the disease ones, they're very skewed. So we've got an excess of very rare functional variants. And of course, we have to sequence deeper into the population to find those. So, maybe that's just all we have, in some sense, what we need to do. We need to sample. But we can think a bit about sampling, and we can try and focus our attention in situations where we can get more strength. And that's really a current state of things. We've got a problem that in populations like most of the world, like Europeans or sort of standard big populations, they've expanded a lot in the last few 10,000 years or so since the Neolithic Revolution. Farming came. So, and what happens in expanding population is that the chance that any two individuals have a common ancestor, or you could share a common ancestor with a sample. So you've got a sample of people we've sequenced. We're going to add one person here. And probably, because the population has been expanding, the chance that there's a recent ancestor is relatively low, and you've got a long final tip length, and on that tip, there would have been mutations which are private to the individual, and so we won't have seen them before. So this means that in big populations, where big is like standard modern populations, the tips are hundreds of generations long. You get tens of thousands of private variants per sample, and hundreds of them are functional. So, is that just life? We have to deal with that? Well, this variation is very dependent on population structure, on this fact that there's been this expansion. If there'd been a different shape of history of population, we'd get a different shape of graph, and that's a sort of standard coalescent graph for people who know about these things. In genetic isolates, the tree relating the different copies of the genome that you have in the sample looks very much more like this, with short tips. And then there's many fewer private variants. That's because you're sharing a common ancestor relatively recently, so you will share something with somebody else in the sample. So is this a real effect? That's a sort of hypothetical thing. Well, we can see it is. So here we're looking at, if you share a common ancestor recently with somebody, then you share a long piece of DNA. If you share with your brother something that's about 50 million base pairs long, the pieces of DNA you share with that individual are 50 million base pairs long. Somebody who you're related to 10,000 years ago, you'll share maybe a 50 million. Somebody 10,000 years ago, you'd share pieces that are much shorter because they'll be broken up by recombination. So those pieces will be a tenth of a megabase, maybe. So here we're looking at a 5 megabase piece, which means it's probably shared about 10 generations ago. And this is within 3,000 British people, taken from the average population sample. What's the chance that you share this particular piece that's 5 megabases long? And it's small, it's just a few percent. Chances are that you're not related closely to anybody in that set of 3,000. If there are 3,000 Finns, there's a half chance that you have a relative in the last 10 generations at this particular place in the genome, in that sample. If you go to northern Finland, which is even more, northern Finland was basically populated in the 17th century by a relatively small number of people who expanded, then the chance is 80%. Then there are extreme isolates. Here we have the Orkney Islands, where there's been a relatively small population. 1,000 years ago, the Vikings came, killed everybody off, founded a new population. Small, relatively inbred, not dramatically, not drastically, but on this scale. So a few hundred people, you have an 80% chance of covering this. And this is an isolate in northeast Finland called Kuusamo, which we're going to look at. Okay, I'm nearly done. Settled by 34 families in the 1680s, current population of about 20,000. It's enriched for certain phenotypes. And we're trying to model this. We think there's a good chance of understanding all the genetic variation in Kuusamo. And then we can expand out from that to populations like the whole of Finland and eventually to our own. So we need a measure of how we can capture the variation in population. And what we're looking at here is a kind of another version of this plot. We're going to take one particular sample size, saying how much can we get with one sample. And here we're saying that 50% of the genome of a new person will be covered in pieces 500,000 long or so. And we've got two lines here, or multiple lines. The green line is, if this was the whole of Europe. The red line is data, so this is real data. The red line is real data from Kuusamo, from this population. And the blue lines are models, simulations of the population structure. 100 founders, no migrations, 12 generations. We can see it's quite a sensitive picture, because if we add 2% of migration, I'm going to stop, then we can fit it better. We can do the same in Orkney, but it doesn't fit quite so well at the other end up there, so there's issues about population structure and history that we aren't modeling correctly. So the real question is, how much variation do we cover with how much sequence? We've had these different ways of looking at it. We're still grappling with that. And it's partly a problem because you can't cover everything except by sequencing everybody. But of course, you can do very well. So in the end, each individual carries perfect mutations. But it is worth thinking about how much you can capture, what are good measures, what are good ways of looking at models, and what are good strategies. Kim Perlin did a lot of the work. I don't know if he's here. I think so. Various other people in the group and the 1,000 Humans Project.